noon here. Just wait another minute just for more participants to attend.
Sure thing.
30 more seconds.
The countdown. Got it, Ross. Thank you.
All right, it is 1 minute past the start of the webinar. It is my pleasure to introduce to you all Brandy Toner from the University of Minnesota. She and I have known each other for a long time, as she's also facilitated my development as a hydrothermal geochemist, and so I greatly appreciate her providing this talk today, which is titled A Deep Sea Tour of Global Hydrothermal Plumes from the Perspective of X-ray Spectromicroscopy. Thank you, Brandy.
Great. Yes, Drew and I go way back, all the way to his grad school years, but I won't tell any stories right now about that. It's been a pleasure to work with you and know you, Drew. Thanks for the invitation. And yeah, let's jump right in. All right. So the work that I'm going to talk about today really is a 15 plus year overview of some of the, I think, highlights of what we've been learning about deep sea hydrothermal plumes. And so various group members listed here at the bottom have contributed to this work over time. But I'm going to be showing some unpublished data as well, and that almost entirely comes from Dr. Sarek Matson, a current postdoc here at the University of Minnesota. And he is now accepted an offer to become an assistant professor at the University of Illinois, Chicago. And so in 2025, you'll be able to find him there. So let's start somewhere comfortable, somewhere I think that we're all very interested in, and just give you some sense that there is now quite a lot of data for globally distributed sites. My group has been visiting some of those sites or collaborating with others to get samples from deep sea hydrothermal plumes for quite a number of years now. And my interest has always been in particle formation processes. And so this really schematic view gives us a chance to think about in the buoyant rising plume, we have a lot of particle formation processes. I'm very interested in that. And I've come to start to think of that as a source signal for hydrothermal venting. I think that phrasing is partially informed by the fact that I do get some NASA funding to think about ocean worlds. I won't talk about ocean worlds today, but this early particle formation process is something I'm very interested in. The other part of my research that I have been following for some number of years is, so you have the source signal, but then there's the transport potential and the signal potentially being altered as it's transported over a variety of length scales. And so this schematic just represents potential biogeochemical and physical processes happening that might result in some different transformed signal upon arrival at a location of interest in the ocean. So I think I saw Jason Sylvan's name on, he'll recognize this photo. So my interest is, can iron get from here, a hydrothermal vent in the deep ocean? What is that signal? And where can it travel to in the ocean? And when it arrives, what is the chemical form that you're going to see? What's the delivered signal for those hydrothermal materials? So just to be clear, pretty much everything I'm going to show today is particulate size fraction. So filtration is a really important part of our field work. And In the talk today, I'm going to try to cover what is the source signal, what is the transport potential, and what is that chemical signature on arrival. Mostly I'm interested in iron, but I will talk to you a little bit about sulfur and carbon along the way. Spoiler alert, there's quite a lot of diversity and variety in that source signal at the hydrothermal vents, if you're very close to the vents. Just saying it's okay to do the captioning. But despite all that diversity, I'd like to convince you that there are some important common themes that we've learned by studying near vent systems, and in more recent years have had the opportunity to do longer transects away from the ridge axis and are finding some interesting themes far afield as well. So my plan for today is super briefly, I'm going to talk about how we get our plume particles. I think of it as going out into the wild and capturing them. And then I'm going to talk about briefly the types of measurements we make to describe the solid state chemistry and speciation of iron and other elements of interest. And then the talk is sort of two parts. There'll be a short part where I'll give you some global summary of plume mineralogy. And then we will move into an exploration of some case studies where we'll look at close to the vent, so within a rising plume, and then a couple of different length scales away from the vent site. If we end up having a lot of extra time at the end, I have some things that I'm thinking about now, a review article, maybe a new proposal. So if there is time in the hour, what's next can be covered too. So here's the brief methods. We get our samples almost any way we can, basically any way we can. If we start on the right side of this diagram, we really like to get our samples through in situ filtration. This work was with Chip Breyer, Chris German, and others, where we can get samples that are collected at discrete locations at known elevations in the rising plume. I'll show you two studies that are like that. Then moving to the left here, we can think about cabled collection of samples, either collecting water and filtering at shipboard or putting filtration devices on the cables. So here on the CT rosette, there's a super sampler, so a filtration device. And here's a more geotraces approach where with Phoebe Lamb, there's Dan Onimus, cabled large platform pumps. If you go back a bit further in history, there's some samples that we worked on with sediment traps at the East Pacific Rise 9 North, or we've also done cabled filtration, again with Chip Breyer. And so I won't talk too much about these two. You're going to see some data from this type of collection in the rising plume, and then there's going to be some cabled sampling for our farther field studies that I'll share with you. All right. One of the things that we spend a lot of time doing is trying to protect our samples from ambient oxygen. So here, Sarah Nicholas is getting ready for one of the geotraces cruises. For those of you who participate in geotraces, this is GP16. And we do a lot to basically get some little particles on filters, and we try to protect those little particles from oxygen. So do our sampling in glove bags and then usually preserve them under nitrogen or argon frozen. Once we've got those precious particles on filters, there is a suite of solid state observations that we tend to make. It's almost like material science with natural messy materials. So we use electron microscopes. Some of these will be very familiar: scanning electron microscopy, TEM, elemental analysis, and also diffraction analysis. So the electron microscopes are great tools. I'll show you some SEM data later in the talk. My main tool for today, and something that I find to be a great discovery tool, a great hypothesis generating tool is X-ray microscopes. So scanning transmission X-ray microscopy, that's a lot to say. We call it STXM, but in this talk, I'll just call it X-ray microscopy. We do this work at synchrotron facilities, and we can do X-ray absorption spectroscopies along with that microscopy. So in my title, when I said spectromicroscopy, this is it. And then we also use synchrotron X-ray microprobes. If you use electron microprobes in your work, it's a very similar idea. It's just the light, is being delivered to the sample by a synchrotron source. We do X-ray diffraction, fluorescence, imaging, and elemental composition, and spectroscopies. I'll give you one example of an X-ray microprobe data set later in the talk. Okay, so let me give you some sense of what's in the literature as of circa 2020 in terms of this global survey. What have we seen so far in hydrothermal plumes in terms of particle chemistry? So I'm going to basically throw some darts on this map. Some of this work is going to be published, and then we'll get to the unpublished data sets a little bit later. So hot pink here, Eastern Laos Spreading Center. We've got some nice published work from Brandy Stewart from my group. Don't worry too much about it. Brandy's not a super common name, but I've had a grad student and a postdoc who are both Brandy. So Brandy Stewart, Amy Gartman did some work there. If you go back far enough, Richard Feely did a lot of really cool early work at Wandafuka Ridge, there in the blue star. East Pacific Rise, Chip Breyer and I, Alyssa Findlay, my grad student, Colleen Hoffman. There's been quite a lot of effort. So the red stars are showing East Pacific Rise, 9 north and 15 south. All right, let's keep going over to the Atlantic. We've got data sets from Amy Gartman at the Mid-Atlantic Ridge. We've got Logachev in orange, published data set, also at Mid-Atlantic Ridge 5 South from 2011. And then let's get into the unpublished data sets. So we have data here showing the yellow star, Mid-Cayman Rise. That's Brandy Crone Cameraman's dissertation work. So we've got quite a lot of unpublished data from that. We also have new rainbow vent field data. That's Sarek's work that's unpublished at the moment, but very close. And a new cruise to Wandafuka Ridge that happened just last September. That's also Sarek's work. I'll give you some little previews and highlights of those. But these two are exciting additions to what's known about hydrothermal plumes because both of them are transects that move between 20 and 70 or so kilometers off axis. So we have these nice transects that we can follow the plume and understand what processes are acting on particles as they move. So this is a InterRidge data set. Adrian Wackett, a former member of my group, summarized this. You've got temperature here on the Y, depth in water depth in meters. And the colored symbols are data sets that we have access to. Either they've been published by us or they've been published by others, or we have them and we will publish them. And then in the gray colors are all these other sites that are available. I think the main thing that I wanted to point out is that he's broken this up by an estimate of host lithology in terms of circles, triangles, and squares. And then also he's basically pointing out the fact that we've got focused hydrothermal venting versus diffuse venting. And the vast majority of the data that we have access to are for focused vents and basalt hosted vents. That's just the nature of the data sets at this time. So I'm going to show you 3 graphs like this. And they take a little explaining. So let me do the explaining now with this one, and then the second 2 will follow, I think, easily. So when you go into the literature, like Adrian did, and you try to summarize all of the particle chemistry, or we could call it mineralogy here because we're trying to name the phases. It's not that easy because the different analytical techniques that I described to you are also used in the literature, and they don't tell you equal things about materials. But Adrian devised a strategy where he basically said, okay, I'm going to take all the basalt-hosted systems, that's 8 vent fields, 16 individual sites, chimneys, and every time I see someone mention pyrite, I'll make a notch. And so the way this works, he's got it broken up by sulfides, oxides, others. Here's elemental sulfur all by itself. All the specific mineral names. If you love mineralogy, maybe you see one of your favorites down here. But I'll just point out that pyrite was found in every data set. So that's what this means. When you've got a frequency percent of 100, it means that all the data sets that he had access to at that time have pyrite. The copper and zinc sulfides were also well represented. Here, something called iron hydroxide. Maybe we can kind of mentally note, maybe that's like a ferrihydrite-like material. Elemental sulfur was also very commonly found. So there's some highlights. So now we're going to look at a different lithology grouping, something he was calling the andesite group. This is a much smaller data set. You've got just 2 vent fields, 12 individual vents, chimneys. And I'll just say that he's organized it exactly the same. The specific mineral names are at the bottom. The big groupings are at the top. Frequency of observation. If it's found in a data set, it gets a point. And so again, for these data sets, pyrite was also observed in all of them. There's less diversity here, but some of the same characters come up: the copper and zinc sulfides, iron oxides, elemental sulfur. And so now if we get down to the ultramafic lithology, as he organized them, there was only 2 vent fields and 10 individual locations where samples were made. We're starting to get pretty sparse on the data, but he did note that pyrite was again found in all of these. But I would say for all 3 groupings, the amount of diversity in the mineral phases was pretty high. So mini wrap-up number 1 here for our global survey of published data available about 2020 is that hydrothermal plume source signal, super diverse. There's a lot of different mineral phases being observed. And there's some common features like pyrite, and so iron, sulfur subscript 2, so reduced iron, iron 3 oxyhydroxides, oxidized iron, elemental sulfurs. Those were some of the common features. And I'll just note again that our data sets are pretty biased toward basalt-hosted systems and near-vent systems in that literature search that he did. So now what I would like to, with that as the sort of reporting out, like where I think we're at, I'd like to do some case studies so you can think about how some of these trends and observations play out at specific field sites. And then also we'll start close to the vent with the buoyant plume, and then look at 2 different studies that follow plumes farther off axis to see how that source signal might be changing. Okay, so we're going to tell you 2 stories today about buoyant plumes, and just kind of think about this, the length scale of interest here is like 1 meter to about 300 meters up into the water column above a known vent. So we're going to go vertical about 300 meters. So the first place we're going to visit is Von Dam vent field at the Mid-Cayman Rise. Some of this work was published by Brandy Crone-Kammermans in 2020 from her dissertation. This was a cool study. There was a lot of sampling where we would get samples for mineralogy, for microbiology, and other observations. And so it's very, very cool study. One of the things I would note is, here's the main vent opening where that star is labeled 1 meter. Here's the little ROV. The vented materials created a kind of milky, hazy sort of plume. In general, people tend to think of this as being a low metals, low particle plume. But there's tons of particles. I mean, there really is a lot. So let me show you what some of that looks like. So here's a little bit of X-ray microscopy where my cursor is and it says carbon image. This is a transmission image. Carbon image tells me what energy of electrons were being, or excuse me, X-rays were being used to image the sample, scale bar 5 micron. Now, if you move to the right, you'll see something called a carbon map. This is an optical density map. So where it's black is very low carbon, and where it's white is high carbon. And so you can see that these little Twinkie features here, once we do the carbon mapping, they are very rich in carbon. Those are probably intact microbial cells adhered to some sort of aggregate. And I'll just say up front that that's a really familiar morphology that we see in hydrothermal plumes in the particulate size class. We get a lot of organic carbon and iron particles. They get stuck together and they travel together. So here's a couple other examples. So take the middle panel. Again, the scale bar is 5 micron. You've got your image here, transmission image, carbon map. That's where the carbon is for the sample. And inside that carbon matrix, you've got a bunch of tiny little iron mineral particles. And you've already seen this panel before, maybe if you've got a good eye for patterns. Scale bar is 2 micron. Here's the transmission image. Here's the carbon image. And what you've got is a bunch of microbial cells adhered to an iron-rich particle aggregate. So this is what we were seeing at Von Diem. Part of Brandy Krohn's work was sulfur speciation. Sulfur is a really great energy source for deep sea microbes. And we wanted to measure the sulfur chemistry in the particulate size class. And so what you have here are X-ray absorption spectra for standards, going from an inorganic sulfate, ester sulfate, sulfone, sulfonate, so forth, green thiol, elemental sulfur, all the way down to an FES type compound like Vigite. So it turns out that if we do linear combination fitting of unknowns to this reference library, we can have a very nice calibration curve. And it's really sensitive to valence state and sulfur. So she applied that technique to particles that were captured on filters for the Von Diem rising plume. And so let me just give you a sense of what you're looking at here. 1, 7, 8, and 86. These are meters above the vent opening on the y-axis. Along the x here, you've got all of these different sulfur speciation types. Metal sulfide, which is a lot of iron sulfide. I'll also point out thiol and organic monosulfides here. Keep going. In red, we get to what she calls the oxidized group, sulfonate, sulfone, ester sulfate, and inorganic sulfate. The size of the circles in this particular graph tells us about the abundance, or like how frequently these different species were detected. First thing, sulfur speciation in the rising plume is really complex. I mean, we're basically getting almost all of the valence states that are available to sulfur in the samples. Also, there's a lot of organic sulfur in these solids, as well as very reduced and very oxidized mineral sulfur. If we take a look just at what's happening at the highest plume elevation here, and you just scan along, you've got your iron sulfides, you've got some thiols, and then you've got your oxidized organic and inorganic sulfurs. So sort of like export signal from this site isn't as complex as the whole sample set, right? As you move up through the plume, things are simplifying. You're getting your iron sulfide minerals forming. You've got some reduced organics and some oxidized organics. That's the source signal from this site. But overall, the complexity is surprisingly high. Pyrite, thiols, and the oxidized forms in red. Okay, so I told you I'd tell you about 2 buoyant plumes. I think we're doing really good on time. I want to take you now to the Eastern Laos Spreading Center. And this is also a rising plume study. It's also published. And in this case, the particulate size fraction was being studied using iron spectroscopy. And what you got here is Chip Breyer's very nice schematic showing the near bottom background and BB, and then an elevation profile and APB above plume background was the vocabulary that we were using. Over here on the right, near bottom background, 1.5 all the way to 200 in the rising plume, and then the above plume background. And then these are reference materials for, well, for visual reference. And so basically, she measured iron speciation for all of these different elevations. And what I'm going to show you, though, this plot looks similar to what I showed you for Brandy Crone's dissertation work. It's a similar kind of plot, but what's included is different. So let's walk through it briefly. So here on the y-axis, we've got the Mariner site, the Kilo Moana site, and the Abe site. And then along the x-axis, what you have is the names of minerals. But in this case, the list is not everything that we observed, but it's the cross-section between what we observed and what can be modeled geochemically by Chip Breyer. So I'll just point out iron 3 hydroxides here on the left-hand side, pyrrhotite and pyrite in the middle, all the way to an iron 2 oxide over here. Now, if you see a gray circle, that means that I wasn't able to detect it with the methods that I use. If it's red, we found it only in the XRD. If it's blue, like pyrite, we found it in XRD data. We found it in the iron Zanes data. It turns out pyrite's actually really easy to identify. It just really stands out, very distinct signal. And then in the green, it means that, you know, here the iron 3 oxyhydroxides were only detected using Zanes because they're not very crystalline, and XRD is not a good way to find them. I'm going to ignore the size for now, just to keep us moving, and point out a couple of things. So for all 3 sites, iron 3 oxyhydroxides were observed. The model, however, did not indicate that they should be precipitating. And at all sites, iron sulfide pyrite was formed. And we saw it with our measurements, and the modeling also detected it. And so thinking about the iron, you know, you end up with a really interesting sort of situation where you get oxidized iron and reduced iron as being some of the main products coming from these vent sites. Okay, so mini wrap up 2. I really like my wrap ups, helps me stay organized. So for the mini wrap ups on the buoyant plume or the rising plume, what we're seeing is that, you know, particles are very much composites of minerals of different kinds and different sizes with biological material and organic material. Sometimes that biological material looks like cells, looks like you know, detritus, like it looks like biota still, but not necessarily living. But there's also gooey matrices that I would say look more like particulate organic carbon or natural organic matter, depending on how you like to articulate that. So this is very common theme. We see this at essentially everywhere we look, that particles tend to be composites in plumes. We also see that in the rising plume, particles have strong chemical heterogeneity, at least for the elements that we've been thinking about so far, both sulfur and iron. And the source signal looks a lot like FeS or FeS subscript 2, so pyrite-like materials, iron oxyhydroxides. So we have a lot of complexity, but in terms of like what I think is actually getting out the sort of the top of the plume, it starts to be a narrower set of materials. All right. So now what I want to do, we've been talking about the rising plume. Now I want to track a dispersing plume or a neutrally buoyant plume. Meg Tivy likes to joke that if it was a non-buoyant plume, it would be sinking. So I try not to say that anymore. So dispersing plume, we're going to try to track it over a length scale of about 100 kilometers and see where that gets us. So that takes us to some new unpublished data. Everything I'm going to show you now is SAREX data in collaboration with Svi Steiner and Eric Ochterberg at Geomar. So there are collaborators on this project. And what they did in 2021 at Rainbow Vent Field is they did a transect. And so this little white box is an inset showing some of the near vent sampling locations. I'll talk about that in a second. Just wanted to point out that what they did is they followed the sort of flow path in the topography near the Rainbow Vent site. And so it's a meandering transect. And so I'm going to give you estimated distances from the vent site. So moving here close to the source, Station 7 is the Rainbow Vent Field. So that's going to be hopefully very clear, very close to the vent or right above it. And then the next site that I'm going to tell you about is Station 12. It's about 20 kilometers away from the source. And then when we move to Station 24, that is the most distant site. So we're just going to look at that axis, 20 kilometers or so off axis and about 70 kilometers off axis. And I'll just say that these are, data hot off the press in some regards. SAREC's done a lot. We're basically done collecting data for the study and are now trying to make sense of a pretty large data set. So here from X-ray microscopy, we have a carbon map. So we've talked about these before. Scale bar is 5 micron. Where the background is black, you've got low carbon. And where you've got white is like a lot of carbon. And so this is carbon map for the sample, one of the on-axis kind of mid-plume depth samples. And here's the iron map. And so you might notice that there's a lot of carbon and not quite as much iron. And it's a little bit harder to tell in this sample. There's, the iron is kind of embedded in this organic matrix, but they tend to be these tiny little particles distributed throughout. So we can use the spectromicroscopy then. So once we've mapped out the elements of interest, we can go in and ask, what's the chemical speciation of carbon? In this case, the carbon is organic carbon. And we can also ask the same thing of iron. What is your chemical speciation in this, in that particle or this particle within our field of view? It's iron 3+. We make other measurements to define the speciation to a greater extent, but iron 3 is the form that these materials are in on axis. If we move about 20 kilometers off axis, I'm going to show you the exact same panels here. We see here's a carbon map from the X-ray microscope, scale bar 2 micron. Here's the same region of field of view for the iron map. You'll notice here at Station 12, we're seeing much more sort of pixel by pixel correlation between the carbon and the iron, so that integration of those materials seems to be more thorough by the time we get to Station 12. But what hasn't changed is the carbon is still organic and the iron is still oxidized at Station 12. So let's do one more. Let's go out to Station 24. Same set of panels. So here's an example of a carbon map. Note the 5-micron scale bar. Again, where it's black, you've got low carbon. Where it's white, you've got high carbon. And here's the iron map. We still see quite a lot of kind of pixel by pixel correlation between the iron and the carbon out at 70 kilometers off axis. Consistent with what we're seeing along the whole transect is that the carbon in the plume is organic carbon and the iron is oxidized. So we've got some pretty consistent trends along that 70-kilometer transect. Now I want to do a little like zoom in, zoom out exercise because we do this a lot in my work, and I think it's really fun. And it really challenges, I think, the conceptual models that we have of what these natural materials are like and how they behave in nature. So let's do a zoom in exercise on the same sample. So this is still the station 24 off-axis sample. And we're using a different kind of X-ray microscope in this case. It's called, and we're using it in a mode called tychography, and it gives us a much higher spatial resolution because you might notice, this looks fuzzy, right? The X-ray microscope that we were using in this case can't resolve the size and shape of the smallest particles. We're seeing them kind of as little clusters of things. But with the tychography, we can get a much higher spatial resolution. At the iron edge, it can be as good as 5 to 10 nanometers. So it's a really nice handoff with transmission electron microscopy. So the color scheme has changed here, so I apologize for that. But where you see dark blue, where my cursor is, those are the little iron hydrothermal plume particles. And so what this does is allow us to see that there are these tiny little nanoparticles. So those iron 3 phases that I was showing you in the past several slides are indeed nanoparticulate iron 3 oxyhydroxides, and they're kind of glued together with organic matter from the plume, and that's a very common arrangement when we zoom in, what we can see. Scale bar, 100 nanometers. So anything smaller than 100 nanometers gets to be called a nanoparticle. We got a lot of those. Okay, now let's zoom out a little bit. Because of course we have many, many samples, many stations, many depths. We want to understand more holistically the system. And so to zoom out, we use a different kind of analytical tool to measure the iron speciation. And so that's now, again, a synchrotron technique, but it's X-ray microprobe. So rather than having one of those images where you've got, a bunch of particles, the pixel size for the X-ray microprobe is basically the same size as one of those microscope images. So we're getting a much more holistic view, and we can do this in a way that, if we're careful, can be quantitative. So let me walk you through this data. So we're going to look at the whole transect now, not just one sample from one station, but the whole transect. These are X-ray absorption spectra at the iron 1s or K-edge absorption spectroscopy edge, for if there's any spectroscopists out there. Here you've got X-ray absorption, and these spectra have been offset to help us view the features. Energy here. We did this work at Brookhaven National Lab. In kind of this greenish color, you've got a reference standard for pyrite. In blue, you've got a reference standard spectrum for the iron 3 oxyhydroxide perihydrate. So all these overplotted lines, those are all of the samples plotted together for the whole transect. And there are lots of things a person can learn about these data just by looking at them. In fact, that's exactly the first thing that we do once they're all curated. But I'll just go ahead and tell you qualitatively what we've learned from this, which is we do see some iron 2 bearing particles close to the vent site. But it turns out that they're not that abundant. And if you take the transect as a whole, they become very rare. So the iron 3 phase is really very representative of this entire transect. So for my mini wrap up number 3, thinking about, 100 so kilometers off axis, and maybe comparing this to what we see right at the vent, we're seeing that particles are still composites in the plume. Minerals associating with organic material is a very common, consistent theme. We saw it near the vent. Now we're seeing it, up to 70 kilometers off axis at the rainbow site. But, our interpretation of what we're seeing in the chemistry of the particles is actually pretty different. We're seeing pretty low chemical heterogeneity. The iron speciation is an iron 3 oxyhydroxide, best represented by something like 2-line ferrihydrite. And it's just really consistently that pattern, the whole transect. I didn't really expect nature to produce something quite that uniform. So it was a little bit of a surprise to me, but this is what we have found. And we've been very thorough about it. So our transported signal over this length scale then is nanoparticulate iron 3 oxyhydroxides. They tend to cluster together and they tend to be aggregated with organic materials. All right, I think we're doing really good on time. I have one more little story to tell you, and then we can have some discussion if people are interested. So now let's jump one more scale, right? So we kind of were like within 1 kilometer, then we were within 100 kilometers. Now let's go to 1,000 kilometers and see what we can learn about the dispersing plume. So some of you will know Colleen Hoffman, perhaps. She was a PhD student in my group, and she graduated a few years ago, and her dissertation was focused on the geotraces transect GP16, which went over the big hydrothermal feature at East Pacific Rise 15 South. So that's what you're seeing here is the cruise transect. Here from Joe Riesing's paper, just showing that the dissolved iron plume, meaning anything that went through a 0.2 micron filter, was large and long-lived, about 4,000 kilometers off axis. From Just Fitzsimmons' work in 2017, Colleen was also an author on this paper. It turned out that the particulate iron signal also had some pretty good travel legs in terms of meaning anything captured, anything bigger than 0.2 micron, so this is all operationally defined, can travel, thousands, 1,000 to 2,000 kilometers off axis, which was pretty exciting to think about the longevity of those particles in the water column. So what are those particles in the water column? That was our job. And so Colleen published some of this work in 2020 in the American Chemical Society's Earth and Space Chemistry Journal. I have to say I'm really liking that journal a lot. Not just because they published Colleen's work, but because if any of you ever read ES&T, which is another American Chemical Society journal, they're really focused more on like environmental quality and pollution. So this is, I would consider like a sister journal where we can talk about chemistry to people who are interested in chemistry, but not about pollution. So it's a good one. So let's go ahead and take a look at some of the data that Colleen got from her dissertation. So from 0 kilometers traveling out to about 2,000. We're going to go from left to right. All right. So these are iron EXAFS data. It's a kind of X-ray absorption spectroscopy. I won't go into the details. If you have an eye for patterns, you might notice some very subtle changes along this stack of spectra going from 0 kilometers, sort of above the axis, out to about 2,000 kilometers. What I'm going to do is take these data and in the next slide give you an interpretation of what's changing with the iron speciation as we move off axis. But it's really hard to resist showing some data. Some, as my spouse says, wiggly lines. All right, so let's walk through it. Here you've got your distance axis in kilometers, and on the Y we've got percent iron 3 oxyhydroxide. We could have presented this a bunch of different ways, but because the chemistry was so uniform, it actually just made sense to plot it in this way. So I'll just point out here, if you are a GeoTraces GP16 fan, here are the stations. 18 was on axis. And from basically station 18 to 26, we found that the iron speciation was super uniform, just like 2,000 kilometers of iron 3 oxyhydroxides in the mid plume. It was very impressive consistency. Around station 28, the iron speciation started to change. We started to see some silicates that suggested not plume material. And then by the time we got to station 30, about 2,600 kilometers off axis, you know, the chemistry had changed to the point where we were pretty sure that most of the iron that we were seeing in that mid plume was no longer of hydrothermal origin. So that's the transect. And it mirrors to a large degree what Sarek is now seeing in the rainbow vent field, which is these clusters of nanoparticles. The scale bar here is 1 micron or 1,000 nanometers. You've got here on axis clusters of nanoparticles that have a lot of organic matter associated with them. We know that from the X-ray microscopy. Station 20 here is about 86 kilometers off axis. You see that same morphology. You've got clusters of nanoparticles, iron 3 oxyhydroxides embedded in organic material. This blank here is just what a polyether sulfone filter looks like. So this very smooth bulbous feature is your filter substrate. All right. So at this point, I guess I would like to compare 3 off-axis, or what we might call far field or distal plume samples. Here from the Wanda Fuka, this is unpublished data by Sarek in collaboration with Chip Breyer, Just Fitzsimmons, and Chris German. This is a filter that was collected from the plume about 20 kilometers off axis at the Wanda Fuka Ridge. Note the 200 nanometer scale bar, clusters of nanoparticles. We've measured the speciation of that iron. It is iron 3+ in oxyhydroxide form. Here, as a recap, here is an SEM. These are scanning electron microscopy, or SEM images. 70 kilometers off axis, we just got done talking about rainbow vent field. That work is also unpublished. 200 nanometer scale bar, clustered nanoparticles with organics. And then Colleen's work at East Pacific Rise 15 South. This is the station 20 that we just finished speaking about. It's about 86 kilometers off axis. And again, I feel like a broken record. Nanoparticles, they cluster together. There's a lot of organic matter attached to those composite particles. So we're starting to see a pretty strong, consistent transported signal from 3 sites that have fairly different initial vent fluid chemistry and fairly different near vent mineralogy when we have that information. We don't have it for all the sites. So this is what I'll call my macro wrap up, in the sense that hopefully it's covering most of what we've discussed so far. So when I think about the deep sea hydrothermal plumes, you know, that source signal is geochemically really complex. There's a lot of diversity of phases. The speciation is a delight to tackle if you're into that sort of thing, which I am. But we can also pull out some common themes. For example, composite particles of minerals and organic materials, super common at the vent site, and still relevant as the transported signal, where we see clusters of nanoparticulate iron 3 oxyhydroxides with organics. So this is what we start with. This is apparently what gets transported if you're thinking about 100 to 1,000 kilometer distances. So I'll finish up here with a working conceptual model that I borrowed from Sarek. And well, you can just consider this working model a work in progress, because I think there's probably some graphics. I think I might have made it more complicated than it needed to be, but let's see. So what you're looking at here is a generic length scale. On axis is 0 kilometers, off axis about 100 kilometers. In this dark purple with a black dashed line, that's representing particulate organic matter. I just wanted to remind you that that organic matter is getting entrained into the rising plume. You can think about lots of turbulence. Those organic inputs we think are mostly coming from the near vent site area. And you can see them here getting entrained, here sticking to minerals, and here being transported away, as well as sedimented to nearby sediments. In the lighter purple, we're showing iron sulfide minerals. Here I'd like you to think about pyrite. Pyrite forms very, very early in the plume. In fact, probably is forming in the vent fluid in the chimney. So you get a lot of pyrite and other kinds of metal sulfide formations. Those particles can get pretty big, have high crystallinity. They're very prone falling out of the water column quickly, but not all of them. Some of them get stuck to organics. Some of them, we have some evidence that some of them start to oxidize. You get sedimented materials. And then I would say some unknown amount of sulfides traveling with organics or as dispersed nanoparticles. That's an open question. There's some evidence that that happens. And then in the brown, we've got the iron 3 oxyhydroxides. You know, the metals in the young plume tend to form minerals with iron very quickly. And then leftover iron tends to start oxidizing. And so these purple or these brown dots are iron 3 oxyhydroxides sticking to organics early in the plume life cycle, being a really important transported signal to the open ocean. And I'll just note that, you know, in Colleen's work on the geotraces transect, you know, the farther we got away from the ridge axis, the more organic material we saw, the less iron. So definitely there was a winnowing of the iron over distance. So this work is funded currently by NASA and the National Science Foundation. In some of our really important method development was funded early on by the Moore Foundation. We've got a lot of unpublished data right now with our GeoMar collaborators, as well as Jess Fitzsimmons at Texas A&M, Chris German at Woods Hole, and Chip Breyer, University of Texas. If you want to talk about this stuff, if you're interested in learning how to make these kinds of measurements, you're welcome to contact Sarek or myself. I have a new grant from the National Science Foundation that is going to include education, outreach, and training materials if you want to get involved in X-ray microscopy. And I will answer any questions if you have them.
That was a great talk. So we have 2 questions right now. The first one was with regards to your carbon and iron maps. Is the intensity scale the same for all map panels?
No, it's not. So that's Donna's question. That's a good question, Donna. What we do is we calculate a 0 to maximum optical density range for each image. And so in the data that I showed you here, I didn't include those, but we do have we do have those calibrated, I guess is the right way to say that. So no, they are not the same for all panels.
The second question is by Steve Goldstein. How do you filter samples to see nanoparticles?
Well, we kind of see them by accident, is the first answer. And then the second answer is with great difficulty. So it turns out that pretty much everything I showed you today came out of the particulate size fraction. And so all of those clusters of nanoparticles are getting caught on 0.2 micron filters. There's good evidence from a variety of groups. Some of that work is published, but most of it is not, that there is also some colloidal size fraction, right? So between 200 nanometers and about 2 nanometers size. And so there must be dispersed materials that are going through the 0.2 micron filters. It's pretty tricky to get enough material in the colloidal size fraction to do our microscopic work. But I will just say that SARC is working very actively on some of those methods with Jess Fitzsimmons' group. And so I'm hoping in the future we'll have the ability to tell folks, what are those phases or species that are getting through to the colloidal size fraction. Because they should be really, really transportable if they don't get gobbled up by an organic glob in the mixing process.
Could you stop sharing your screen so we can share the video of ourselves?
Yeah. Let's see. There we
Perfect.
go.
Another question by Steve was, could you use this technique to ID unknown off-axis vents?
Well, we've kind of gone back and forth about this. If you grab an ocean sample and you see the chemical speciation of iron and carbon and the particle morphology that we're defining here, it would be suggestive of it being hydrothermally sourced. But I think probably the other methods that folks are using to track plumes, the other sensors that people are using to track plumes are how we're confident that our particles came from a plume. So no, I don't think the microscopy in and of itself is standalone good way to track plumes. That's my first answer. I guess I'll think about it some more.
Another question by Donna is with regards to sulfur speciation. C reduction with height of metal sulfides and increase in inorganic sulfates. Is this reaction of metal sulfide particles in that short space of time? Are they being oxidized? If I stated that question properly.
Yeah, I'm looking at it too. I can see it in the chat. So I think, Donna, what's happening at Mid-Cayman Rise is, there's some mineral sulfate in the Mount Dent structure. So I think some of that particulate inorganic sulfate is actually just getting lofted up from the mound. The sulfone, sulfonate, ester sulfate, that could very legitimately be part of microbial biomass or other kinds of, there's a lot of vent shrimp at that particular site, and we can definitely see their chitinous material in the organic carbon spectra is a strong source. And so I think the, Mount Dent, the mound and also the vent fauna are contributing to the particle characteristics. And I would say that, yes, we do, in addition to that, we do think that some of the sulfur is getting oxidized in its travel path from like 1 meter to 70 meters or something like that. But not enough to erase that FES signature.
A question by Jason Sylvan. Does your recent work indicate to you that any of this iron may reach the surface in parts of the world?
Yeah, that's a really good question. I think the optimism that I hear from oceanographers about this waxes and wanes. I guess from my point of view, I think the answer is I don't know. I feel like it's really a modeling question at some level, right? Like, how do we, if we do a transect through a long-lived oceanographic feature, we can learn things. about how far can particles travel if they have certain characteristics. It turns out they can travel thousands of kilometers. So, does that water mass eventually upwell? That would give you your transport mechanism. It's my understanding, Jason, that the Southern Ocean is also thought of as one of the places where this might be important in a direct way. But I've kind of kept my research on the side of like, what's the source term? What's the transport potential? And I'm hoping that somebody else is going to pick up or continue to pick up the modeling components. If you know what a particle is made out of and how it's constructed, should be able to put it in a model and try to understand where it can go with different physical oceanographic conditions. So
We have time for just one quick question, and it's by me.
okay.
Do you also look at the trace element geochemistry of those particles, like say chromium or
Mhm. Yeah, always, but always through collaboration, Drew. Yeah, we work with, Eric or Jess or Chip Ryer to make sure that the trace elements get measured and measured really well. And, sometimes isotopes are part of that suite. Just depends on the collaborative group that we're interacting with.
Great, thank you. So I believe we are at time. So thank you for your talk. It was great. Really appreciate you.
Thanks, Drew. Thanks, everybody.
Take care.